This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 17 for January 23rd, 2009. I'm Vincent Racaniello.
I'm Dick Despommier.
And we are joined today by Saul Silverstein. Welcome, Saul. Welcome back.
I'm happy to be here.
Saul was on TWiV previously. He's my colleague, our colleague here at Columbia, right?
Exactly. Oh, we are his
a world-class, well-known virologist.
Exactly.
Who will help us talk about great achievements in virology today. One of our listeners a few weeks ago asked us to do this, so we're doing it for our listener. How about that? We respond. But before we do that, Dick, I wanted to ask you, do you have your TED Talk ready?
Yes and no. I have the first version. Actually, the way these things work is they subcontract to a PowerPoint massage group. In this case, it's called DoArt, D-U-A-R-T-E. They're out in California.
Wow.
And they actually place your talk into their format so that everybody's talk looks basically the same in terms of fonts and colors and backgrounds and things of this sort. But all of the slides that you send them, they then remake that data. So I'm in the phase 1 of this. I'll be in phase 2 this afternoon when I get back to my desk. And by next week, I should be in good
Hopefully
shape.
we'll have a successful phase 3 and we'll be able to market it.
This is all true.
They're very controlling, aren't they?
They extremely.
So they want a really consistent product.
They
right?
do, and they get it, too. They're very, very thorough. In fact, they even have rehearsals for this, and it's a very involved deal.
When is the talk? What day?
Well, it's like showbiz. It's on February 6th.
So will you practice it for us ahead of time so
I'd
we
love
can
to.
hear? Because we can't afford to go. What is it?
No, if I was just going to listen, I couldn't either.
It's $15,000 a ticket or something?
It's a big price.
I'd love to go, but you're going to be rubbing shoulders with Bill Gates.
I am. And Oliver Sacks. Don't forget Oliver Sacks.
But speaking of Bill Gates,
yes,
and one of our two stories today,
yes,
Gates Foundation pledges an additional $255 million to eradicate polio.
Wow.
Going to take care of the Indian problem.
Can they do it?
No.
Well, you know...
So you say no? Vince, what do you say?
Yeah, eventually, but not in 5 years, which is what this money is. So they say that this money is to finish it off over the next 5 years, finish the eradication. I think that's a bit optimistic because we're 8 years behind schedule already. Right. But let's look at these numbers here. So Gates, they have raised a total of $6.17 billion so far for eradication.
Wow.
And the biggest contributor is...
World Bank?
World Health
No.
Us, the U.S.
The United States.
Also, the Gates Foundation has contributed quite a bit, $300 million this last round. Germany, Britain, and probably other countries. It all goes through Rotary International.
Warren Buffett?
I don't know, maybe.
Indirectly because of the money that he gave to Gates to spend for him.
Exactly. I wish you'd asked me first.
So, all right, here's the story. Gates pledged $100 million in 2007. And then he just did another $255 million.
That's remarkable.
And Gates says, if we don't do this, we will lose all the investment we have made in the past.
Wow.
He's very, very, very right on that. The problem is that in areas such as India and the mountainous Himalayas and Africa where you cannot get complete protection, and we know that there has been disease, and we know that there are carriers who continue to shed virus, we have a long-standing problem. Vaccination has to be not complete, but certainly you have to have enough people vaccinated so that you get a real herd effect.
Right.
And not being able to reach people because you don't know about them or because they are not informed about the possibilities makes this an incredibly difficult problem.
Exactly.
So what was the trick about smallpox then? We did get rid of that. So why is that different than polio? As a non-virologist, I'm allowed to ask that question.
The difference is that every infection with smallpox gives disease that's quite visible. So whenever there was a case of smallpox, they would rush in and immunize the village, and that would stop it. With polio, 1 in 100 is visible.
I should comment that I asked that question knowing the answer already, but I wanted our listeners to have the advantage of having it asked for them.
But there's more to it than that, and it's really important to understand that in that population of people who are infected without apparent disease other than perhaps a cold, you have shedders.
Right.
And shedding is a disaster, especially now that we know from all the beautiful work that's been done that the virus reverts in these shedders, and it becomes no longer a vaccine strain, but rather a pathogenic strain again.
Sure.
And that's a real issue.
Yeah, and it's long-lived in the environment after it's shed also. Which
It's
is
a happy camper, isn't it?
Yeah, it is. It's passed along from individual to individual, so it's a real problem. At some point, as we've said before, we have to switch from this live vaccine to a killed one, but we have to get there first. And then, of course, the other problem is you can't stop immunizing, as happened in Nigeria. The government decided, well, we don't want to use this anymore.
Well, look at measles.
That's another problem, too.
The Brits stopped vaccinating, and they've had a scourge of disease.
They have a huge They have outbreaks in Switzerland, the UK, and other developed countries,
like Germany,
because people are not taking the vaccine. There's this parental backlash against being immunized, and that's taking its toll.
Right.
Kids are getting sick. Does this make sense?
No.
Doesn't make sense at all.
Nope.
Now, another polio story, which is very interesting. I just saw today that the world's first bivalent polio vaccine is being... you sent it to me, Dick.
I did send that to you.
Thank you, Dick.
That's quite right.
So the oral vaccine that Albert Sabin... Let's go back in history. Albert Sabin developed the oral vaccine. When it was first used in the U.S., you actually got 3 doses, type 1, type 2, and type 3. So you had to go back 3 times. And then they found, for a variety of reasons, it was better to combine them all and give what's called a trivalent dose. But you needed 3 of those because 2 of the 3 viruses wouldn't grow when they got into your gut after you ingested the virus. Type 2 was eliminated a while ago from the globe. So in India, for example, they were using monovalent type 1 and monovalent type 3 separately. So you had to go back at least twice.
And I told you my story. I just got back from India, so they actually marked the doors with what the strain was they gave, when they gave it, and to how many kids. So they are keeping track, but the infrastructure might not exist at the foot of the Himalayas. You're absolutely right, Saul.
So this makes it more efficient, giving the types 1 and 3 together, but it's never been tested. So they're actually testing it in India by giving it to kids and then looking at their immune response. So they have to take blood and measure antibodies. So it's a little clinical trial. I don't know who made this vaccine. If it's made elsewhere and mixed, or I couldn't find any information on that, but maybe we'll find some in the next couple of weeks.
Well, let's hope that the Indian pharmaceutical industry got involved. They have tremendous capacity, especially when it comes to medicinal chemistry and synthesis. And this would be a wonderful opportunity for them to expand that into the vaccine area, especially as it relates to protecting the subcontinent.
Yeah, they are very good and inexpensive, right?
Very inexpensive.
All right, gentlemen, would you like to talk about the great achievements in virology?
Go for it. Absolutely.
All right, as I said, one of our listeners asked for this, and I thought it was a good idea. We have a list of them, and as if either of us have something to say about it, we're going to go way back to 1796, which I don't remember.
That's even older than I am.
I'm disappointed. I thought you'd be aware of that. But you have to go back before that.
Uh-oh.
You have to go back to the ancient Greeks and Romans and Herpes gladiatorium.
Tell me about that.
Well, there were all these little vesicles and rashes that kept appearing on the gladiators. And it turned out that close, intimate contact that you have when you wrestle was spreading virus between the warriors. And this was some of the first instance of virus spread.
So we have this recorded?
We have this recorded, and that's where the name comes from.
Of course, they didn't know it was a virus.
No, they didn't know it was a virus.
They didn't even know it was an infectious disease.
But then again, neither did Jenner.
Yes, no. Jenner didn't. So the first achievement is Jenner, right, who made a vaccine against smallpox.
Sure, but you won't count the Chinese in scarification.
Ah, of course.
Yeah, same
Before
thing.
that.
Same game.
So that was a sort of a folk medicine hand-me-down cure, and they did that for leishmaniasis also, which is a protozoan infection. It works for that as well. So, as soon as an organism becomes attenuated during the curing period, as a patient's immune system catches up, that's the time to pick the organism to try it out on somebody else.
But you've got to be lucky.
You do have to be lucky. It has its caveats and drawbacks. There's no question.
Yeah. I mean, do that with smallpox, you could end up with a lot of
you're right.
Dead phase 1 patients.
That's true. That's true. The failed clinical trial number 1.
Dick, you're absolutely right. The Chinese were doing smallpox scarification long before Jenner, and maybe he had heard about it. Perhaps. But Jenner, of course, noticed that milkmaids who got cowpox had milder smallpox.
Famous story.
So he took the pustules of cowpox and immunized a small boy, and he was protected. First clinical trial? No, the Chinese probably did it before that. Jenner, and then the procedure was called vaccination by Pasteur in honor of Jenner, because he used cowpox, vacca vaccine, right? So 1796. Pasteur made a rabies vaccine in 1885.
Yep.
So that's a big date. But we must remember that we didn't, just in 1885, we had the concept of infectious diseases by then, because Semmelweis, Pasteur, Koch had all established this idea. But Jenner didn't really have this, maybe mildly. Remember, it all began with Lewenowik, with him seeing small things, microorganisms, and from then it built. And so Jenner, it was not very strong, but certainly was with Pasteur. Sure. But they didn't understand viruses yet.
Nope.
They knew infections and infectious organisms, but not viruses.
You can't leave Metchnikoff out of that either.
What did Metchnikoff do?
Well, he was studying these little water fleas and noticed that they developed an infection that could kill off some of them, but their cells would ingest them and protect them from it. And so if you go to the Pasteur Institute in Paris, at least, there's a big portrait of Metchnikoff sitting right up on the top.
Next time I'm there.
Yeah, it's a great place to go to, by the way. For me, that's Mecca.
All right, so this brings us to the first identification of virus, and that's at the end of the 1800s, 1892 and 98, 2 independent observations. It was a disease that was very important at the time, tobacco mosaic disease.
It was killing our livelihood, Sir Walter Raleigh said.
Kill them one way or another.
That's
right. Two groups, Ivanovsky said you could filter an extract from the diseased plants, and if using a filter that would hold back bacteria and transmit the infection. So he said, this is interesting. This is an infectious agent. Right. But that's all he said. Now, Bayer Inc., who did it 6 years later, did the same experiment. He said, this is a novel agent because it's really small. Okay, so the first guy didn't quite get it, Ivanovsky, but they're both credited with discovering viruses.
They did the right experiments.
So the filtration business defined it, although I understand that a virus was actually used to describe all infectious agents at the time. It's from the Latin word meaning poison.
Sort of like the vapors.
Vapors, yes.
Yeah.
So, but they were the first to do the experiment, and that led to 1902, the first human virus, which is yellow fever. Walter Reed.
Called that the Panama Canal virus.
That's right.
1908 polio virus. So those are 2 important human viruses.
And this is Landsteiner, the same guy who did the blood group work.
It is.
So immunochemist.
Yeah. It's always amazing how when you look back historically at the breadth of some of these scientists.
We had a direct descendant of that lineage here. Landsteiner trained Heidelberger, and Heidelberger trained
Cabot.
Cabot, how about that?
Cabot trained us.
And I knew Cabot, yeah.
Yeah, we all did.
Okay, so that Landsteiner was a pathologist in Vienna, right? Then at the same time, 1908, chicken leukemia virus by Ellerman and Bang. Make a cell-free filtrate extract, put it into chickens, they get leukemia. Followed in 1911 by Rouse sarcoma virus, Peyton Rouse.
Showed transmission of a filterable agent that caused tumor genesis.
Right. Yep. Solid tumors, right?
I actually met him.
Where was he at Einstein?
He was at Rockefeller.
Rockefeller. Yeah. What a name, Peyton. Peyton Manning.
Right. Rouse wasn't as good a football player, but he was a better scientist. So it's all right.
1915, viruses of bacteria, bacteriophages.
Now there it is.
Twort and Darrell.
The Jonathan Swift quote immediately pops into my head. That is, little fleas have smaller fleas upon their backs that bite them, and smaller fleas have littler fleas, so on, ad infinitum.
It's great. That is great. I remember that, yeah. And then 1938, the first virus vaccine where we understood what we were doing, as opposed to Jenner and Koch. Pasteur.
I would argue with that statement.
What would
I
you say?
still don't think we understand what we're doing.
Okay, that's a good point.
The complexity of the immune system and the exquisite
You're right.
sensitivity that it displays for dealing with invasive agents, everything from the innate immune response to antibody and cell-mediated immunity and so forth and so on is remarkable. And we don't really understand to a large extent what mediates immunity and immune
You're right.
response. And that's,
Yep.
and
what
we keep
doesn't
guessing.
immune-
And what doesn't work? What doesn't work and why?
Why some people get sick and why some don't. And that's the whole crux of it. And you're absolutely right. And what is a recovery period and all these other things that and transmission and carrier states? It's an infinite shade of gray. And we only know the outskirts of that. We know the extremes, but we don't know the middle part.
Nevertheless, what I had in mind was yellow fever vaccine,
Okay.
1938 by Max Tyler, who was at Rockefeller also.
Tyler
correct?
virus.
Tyler's virus, a contaminant of the mouse colony at Rockefeller.
Or everywhere else, as the case may be.
1938. And then, same year, electron microscopy of viruses.
Wow.
Von Boris, Ruska, and Ruska. And they were the first to see pictures of these things, so you could tell what they looked like, how big they were, how small.
And the pictures were incredible. If we only had Sputnik before that, we would have had an idea of what we were
That's
looking
right.
at.
That's right. And this, of course, is happening until this day, electron microscopy. 1939, this is a big one, one-step growth cycle.
So important in our beginning to understand what it is that a virus does when it enters a cell, and the fact that they replicate even though unto their own they're not alive. It sort of reminds me of my college biology test when I had a professor who walked into the room, lit a candle, and said, "Is it living or dead?"
Excellent.
Final exam.
What was the
No
answer?
kidding.
The answer was what you wrote.
There's no answer.
And it was great.
As long as you wrote something, right?
As long as you wrote something.
That's right.
Well, Ellis and Delbruck did that. They took phages, they attached them to bacteria in a way that synchronized the infection, and then they let the infection go and took time points and measured the amount of virus produced. So they could see, as Saul said, the virus went in, infectivity went away, and then it would slowly increase.
Vince, of the ones that you've already read, how many of those were Nobel Prize winners, if I may ask? Because I know a lot of them were.
Well, Peyton Rouse was recognized 90 years or 80 years after
a long time.
discovery.
That's right.
And
I can tell
did
you.
Landsteiner win a Nobel Prize?
No, he didn't.
I don't
No, he
think
did
so,
not.
but he
He
should
did
have
not.
for the blood chemistry
That's
work,
right.
which is pretty remarkable. And I
All right, of the ones so far, the Nobel Prizes are just Peyton Rouse, right?
Yeah.
That's remarkable.
But we have some yellow
Well,
fever
Delbruck.
vaccine. Sorry, yellow fever vaccine.
right.
We have one-step growth cycle was a...
It was Delbruck, Delbecco, and
No. You're thinking
that's
of
the other one.
someone else.
That's cell culture.
You're thinking of Delbecco,
That's the Weller.
Baltimore, and...
Yes, I'm sorry. Did Delbruck win a Nobel Prize?
Of course.
Yes, he
Yes,
did for this one
Luria
step.
and Delbruck.
That's right.
That's
That's
what
right,
it
Luria
was.
Delbruck.
You left Luria.
So we have some others coming up. We'll point out when they get prizes.
Yep.
1941, Georgehurst, first virus-associated enzyme, neuraminidase of influenza virus. So the idea that the particle could actually have an enzyme in it. And he was here at the Public Health Research Institute in
Downtown
New York. Yeah.
And that's really interesting. Remember the other day I showed you that article about resistance of the neuraminidase to common
Antivirals. Yeah.
now. And that resistance goes back to the swine flu virus. And it shows us that the importance of structural biology as a contributing tool towards our understanding something about virus mechanics and the development of antivirals.
Absolutely.
And antivirals are something that we still have yet to come to grips with in a big way.
Yeah.
I
If you
think
don't
that's
understand that they reproduce, how can you interfere with their activity?
well, but
We don't know all the steps. We don't know all the chemistry involved.
no, but we've been very successful, and one of the things that I would note before we go much further is we've failed to identify in this wonderful list of important discoveries in virology the development of the antiviruses or the antiviral agents by and I'm blanking on her name, Gertrude Elian, and I can't remember the
What
other.
year would that be? I thought they began in the 60s.
Yeah,
mainly.
but they really began early.
Acyclovir, amantadine, those were
Right,
the
all these
earliest.
guys were.
Yeah, they were looking.
We're looking.
Oh, the sulfonamides, which were the antibacterials, that's 30s, 40s.
Mhm.
That really stimulated the antiviral programs too. Yes, that's a good
And
point.
and the antibacterials, of course, were as important for dealing with infections as they were for the development of cell culture.
Right.
we would have no
That's
cell
right.
culture
Yeah,
without
exactly.
antibiotics. And antibiotics, this is how Enders and Weller were able to do their first virus growth studies in mammalian cells.
Absolutely.
We had antibiotics.
And you wouldn't have a molecular biology without viruses and bacteria.
Of course.
So
No, that's a good point. In fact, the one-step growth cycle, this was, Max Delbruck was a physicist, I think.
Yes.
Going into this new area, and he recruited a whole slew of physicists who thought very analytically, and they went on, Seymour Benzer,
exactly, he was
to break down the gene and understand what it was.
Yeah.
1948, now here's the polio replication in non-neuronal cell cultures, Enders, Robbins, and Weller. That was a Nobel Prize.
Right,
First
and then
time.
Weller subsequently went out and did some of the really important work on varicella zoster virus very shortly thereafter.
And of course, that growth in cell culture, people could not grow viruses in cells before that. They had to grow them in animals. And as you said, the antibiotics
Embryonated eggs
made it possible.
exactly.
And here's another big one. So that got a Nobel Prize. Here's another big one. Plaque assay by Renato Dubecco.
Oh, wow.
And he did a plaque assay for polio. He actually took the procedures that the bacteriophage guys had developed for titrating phages, and he applied it to polio.
57 years ago. It's remarkable. The most remarkable thing is it's still the most frequent assay we do in the laboratory.
I love it. It's a great assay.
It answers so many questions.
Yes. 1952. So Dubecco ended up getting a Nobel Prize later. Probably that contributed to it. 1952. I'm missing something. Oh, no, it's the same year. Viral genome is nucleic acid. The famous Hershey-Chase experiment. You take a bacteriophage, right? You label it with P32, and that the nucleic acid gets into the cell. You label it with S35 to label proteins. It stays on the outside. That was a key experiment. Now, that was a Nobel Prize winning experiment also. If they had used the phage that actually, where the whole thing got in the cell, which we know happens nowadays, they would have gotten a different answer.
That's right.
But sometimes there is, what's the word? Serendipity. Thank you.
Exactly.
Serendipity involved.
Pasteur says luck favors the prepared mind, and in this case, you have to know when you've been lucky.
And luck favors the lubricated mind also. 1954, Salk's inactivated polio vaccine was released. He didn't get a Nobel Prize for that, but we should note that in 61, the oral attenuated vaccine of Albert Sabin was released.
I didn't realize it was that long after.
Yeah, I mean, Sabin was working on his in those years, but yeah, it was a long time afterwards. And that's an interesting story in itself, which we'll do another time. 1957. Now I'm 4 years old. In vitro virus assembly, Frankel, Conrad, and Williams, they took RNA and protein of tobacco mosaic virus and mixed them together, and they made infectious virions. And that's an important conceptual advance, self-assembly.
Right.
And the tobacco mosaic, remember, that was the first virus identified.
Right.
In fact, it was the first one crystallized. Now it's the first one assembled. So a lot of these early advances due to a plant virus. In the same year, 1957, interferon was discovered. Isaacson Lindemann. Although I've heard people disagree with them having discovered it, but I don't know the alternative.
I don't know what anybody else
Interferon
either.
is huge, of
We're
course.
going to hear from our listeners, I'm sure.
Oh, by the way, if we miss things that you want to hear, tell us, yeah. 1961, Kasper and Klug elaborated the principles of icosahedral virus structure. So they had been seeing these electron micrographs of particles, and they had icosahedral symmetry. And by this point, we were also starting to analyze the proteins in particles. And they said, an icosahedron. These proteins are arranged with this kind of symmetry. It gives you rotational axes of symmetry, and it's the best way to build a particle from a few proteins.
And Buckminster Fuller would argue that it's probably the best way to build a house, too.
Or a soccer ball.
Or a soccer
I'd
ball.
like to add a name here, although I'm blocking on the name. I'm not blocking on the result. He was a medical student here who took a medical elective in the tropics in 1951. He went down to Colombia, South America, and discovered a subset of people who were not infected with hepatitis, but they had a certain antigen which was circulating in their serum, which matched with the hepatitis virus. This was in the early days of discovery of hepatitis. He then went to Australia.
This is Bloomberg. It's coming.
1963. It's next on the list.
Yeah, but that's 1951. As a medical student, he did this. That's where he got his idea from.
Well, he followed it up.
He certainly did.
This is Bloomberg?
He certainly did.
Sometimes it takes a little longer than other times.
So Bloomberg was a medical
He
student
was a medical
here?
student here and studied with Harold W. Brown, who was my mentor, and was sent down. He could pick any place he wanted, so he went to Colombia, South America, and got involved in hepatitis. And okay, I'm sorry to have spoiled that.
No spoil at all.
Because it actually began in 1951. I noticed that you had it in 1961.
1963, we have the Hep B
63.
virus discovery by Bloomberg, but that's fine. It's
Sure.
beautiful background information, absolutely.
And it suggested that it causes cancer, and that prevention of hepatitis is a cancer prevention. I also don't see Shope on that list.
Yes.
You should see Shope in there someplace because of the discovery of papillomavirus, but also because of his investigations in swine influenza and proving that it's transmitted from pig to pig by the egg of Ascaris lumbricoides, which is in my bailiwick, a parasite as big as your pin.
And that was in the 30s, I think.
That's right.
The Shope and the flu work. Yeah.
That's exactly right.
Well, I took some liberties.
It's okay, Vince. We're here to fill in the gaps.
Yeah, fill in. Next one is 1964. The first human tumor virus, Epstein-Barr virus. Epstein-Barr and Burkitt. Maybe Saul has some insight.
I don't have a lot of insight to this, but the occurrence in these children in equatorial Africa with the development of this tumor and tumor mass in the face was analyzed in great detail by Epstein and Barr. And Burkitt, I believe, had actually done the epidemiology, if I have it correct. And Epstein and Barr actually were able to propagate it and see it by electron microscopy. And that goes back to the statement you were making before about how valuable it is to see things.
Right.
And that enabled us to associate this virus as a member of the herpes virus family.
Yeah,
and that was one of the first major thrusts towards looking for the roles of viruses in cancer. So we had the hepatitis that Dick had spoken about before, and now Epstein-Barr virus. And at that point, the world went crazy, and we started to try and associate every virus we possibly could with some form of cancer. And there was some success.
Sure, absolutely. What was the manifestation of Epstein-Barr virus in non-Africans?
Mononucleosis.
There you go. So immunogenetics plays a huge role as to whether you get something or something else, right?
Well, we don't really understand it because EBV causes 3 very distinct diseases. The first is the Burkitt's lymphoma, solid tumor. Second is nasopharyngeal carcinoma, which is pretty much
restricted,
to Southeastern Asia. And what the basis for that is not clear. And then, of course, there's the mononucleosis, which is a worldwide B-cell infection.
Now, do you find that in areas that also have Burkitt's lymphoma? Or
I believe you find it everywhere.
You do? Okay.
Yeah. Okay. So that's an interesting issue. And the virus lays there and it's dormant and it does very little. And then sometimes it causes a lymphoma.
Exactly. And herpes is, and you're certainly more aware of that than I am.
Yeah, and it's treatable. It's treatable with these antivirals that we were talking about before.
Okay, 1966, the Kuru story of Guy Dussek, which we talked about because he died a few weeks ago.
He did.
He was the first to show that these spongiform encephalopathies could be transmitted in humans. We knew about it previously in other animals, but he showed it with the 4 tribe in New Guinea. We talked about that. So, 1967, the first virion-associated polymerase by McCausland and Cates, vaccinia virus. They showed there was an RNA polymerase, a DNA-dependent RNA polymerase, actually in the particle.
It made so much sense, too, because the virus was restricted to the cytoplasm of the cell, although there is a very bizarre nuclear requirement for vaccinia replication. But one always wondered, well, you've got this DNA genome. It's sitting inside the cytoplasm. How does it make RNA?
Right.
And there was the answer.
It was.
What about Hillary Koprowski and Michael Katz, who are rescuing measles virus from SSPE victims? Does that count?
Sure, you can bring up whatever you want.
No, I'm not sure how major accomplishment that is, but I think it required a heroic amount of work and tissue culture.
So the fact that,
of all of these
in other words, the fact that SSPE is a late sequelae of measles, that's important.
Yeah.
I don't know the year of that. We'll
It
dig
was in
that
the
up.
60s. I know it was in the 60s.
By the way, we've neglected to say that Bloomberg got a Nobel Prize for Hep B.
Just happened in passing. So.
And the next discovery is another Nobel Prize, 1970, retroviral reverse transcriptase.
And that blew the world apart because
Temin
in Baltimore's discovery of reverse transcriptase led to a whole field of developing ways to associate retroviruses with specific infections. Having this exquisitely sensitive assay that was specific with some caveats led a large number of groups to look at various human tumors and try and identify retrovirus particles associated with them. And unfortunately, most of that work was bogus.
Oof.
And what else did it lead to?
2 Nobel Prizes.
Led to 2 Nobel Prizes and great tools for molecular biology.
Yeah, for cloning, right? Recombinant DNA. And that's
absolutely.
our next. So they got a Nobel Prize for that. And our next one, 1972, Paul Berg, the first to do recombinant DNA, put a viral genome into a bacterial vector.
Right.
He puts SV40 into bacteriophage lambda. And our colleague next door, Steve Goff,
did those experiments.
Yes, he was involved in
What
that.
does SV stand for, Vince?
Simian virus.
Thank you, Vince.
You're welcome.
Vince has a double voice here.
No problem. 1973. Here's another Nobel Prize-winning discovery. Viral antigens are presented to lymphocytes in the context of the major histocompatibility proteins. You remember the guys who did that? Toherty and Zinkernagel.
Zinkernagel. You're absolutely right.
Yeah, Nobel Prize. Really important stuff.
Yep.
And only the beginning. 1974, the first transgenic mouse, Beatrice Mintz, in Philadelphia, put SV40 into mice.
I worked with B on a project to express globin genes.
Really?
Yeah.
Wow.
And they were the only people who could do it.
Wow.
Injecting these things into cells that became mice.
Now everyone can do it.
High school students do this.
Actually, now you pay to have it done. You don't do it.
It's a lot easier.
In fact, like many of these technologies, you pay now to have it done. 1975, Aaron Shatkin and Bernie Moss discovered a very unusual structure on the 5' end of the mRNAs of reovirus and vaccinia virus, and we now know that's a cap, an mRNA cap. Ushered in a whole era of understanding how mRNAs are translated. 1976, here is another Nobel Prize-winning discovery. Retroviral oncogenes are derived from cells, Bishop and Varmus. Important
Varmus,
stuff.
another PNS medical student.
That's right.
Should mention those things.
Here in New York, of course, and he's also Obama's science advisor now.
Oh, wonderful.
1976, the first viral genome sequence was a bacteriophage, MS2, by Walter Fears. It was a big deal. And here we go, 1977, Nobel Prize for RNA splicing, Phil Sharp and Rich Roberts.
That was an amazing time.
Wow.
And splicing had tremendous ramifications for how virus genomes are used. And I remember very distinctly at this time I had invited Tom Broker to come to Columbia and give a talk. And he was, I don't remember what he was working on at the time, but when he walked into my office, he said, "I'm not talking about that." He said, "We found this really amazing thing." And he was then at Cold Spring Harbor, and he had collaborated with Rich Roberts and that group. Roberts was another one of the people who shared in the Nobel Prize, as you've mentioned. And they started to talk about adenovirus splicing, and Tom had done the electron microscopy to show the splicing intermediates. And it was one of those seminars where you walked in and you walked out and you realized that the paradigm had shifted. And it was really amazing. And it was subsequently followed by the work of Leader on globin genes and O'Malley on ovalbumin, showing that this is a general phenomenon, one of many.
Wow.
I remember I was a graduate student at the time, and my advisor, Peter Palazzi, had gone to a meeting and heard about this, and he walked in the door and he said, "This is the next big
Stop
thing."
Everything.
Now, we worked on influenza virus, and the conventional wisdom was, well, it's an RNA virus. It's not going to splice, because here there was splicing going from DNA to RNA. But many years later, splicing in RNA was also found.
Sure.
And here we are, 1977. Axel Silverstein and Wigler. Would that be you? Saul
that'd be
Silverstein?
me.
Really?
DNA transfer into mammalian cells.
Wow.
Which has also allowed us to do many, many things, not only in research, but in practical matters as well.
Was that a baculovirus type thing? Or
No, that was a the question that we asked was, could you take a virus gene that expressed an enzyme that we understood something about, thymidine kinase, and transfer it into a cell? And we had then tools to begin to pursue this problem. We had cells that lacked thymidine kinase. We had selection pressure that would identify cells that incorporated and expressed a gene of interest. Mm-hmm. And we were able to proceed and isolate the gene from herpes simplex virus that encoded thymidine kinase. And that really began a whole new field of mammalian cell genetics, and it provided the basis for a pharmaceutical industry to produce proteins in animal cells. Right. And it was a lot of fun. We had a good time doing it.
One of those guys got a Nobel Prize, but not for that work.
Not for this stuff, though.
Richard Axel got it for scent receptors not too long ago. 1978, the first infectious clone DNA copy of a viral genome, Q beta, a bacteriophage, by Charles Weissman, allows manipulation of RNA viral genomes.
I noticed that you've left out the similar finding in animal viruses.
Mm-hmm. And what would that
Dr.
be?
Dr. Racaniello.
That would be the first infectious clone DNA of an animal virus.
That
right?
would be you.
Okay, I can't put it in myself, but since you mentioned it, that would be 1980.
Tip of the halo hat to you, my boy.
Racaniello in Baltimore. 1978, the first virus X-ray structure by Steve Harrison, tomato bushy stunt virus. Another plant virus leading the way.
Easy to obtain. Right?
Huge amounts.
Yes.
Lots of economic loss as a result of encountering them. So.
Huge quantities and computing power now at a level where we can solve these structures, because this is not just globe and a small protein. This is a huge assembly, and you need intensive computing. 1979, smallpox eradication. Important.
Very.
The work of many people. 1981, the first human retrovirus, HTLV1. Human T-cell lymphoma virus, Bob Gallo. So he did discover this virus.
He discovered this virus, and he certainly developed IL-2, which was a critical reagent for propagating cells that would allow for replication of this virus and for HIV.
1982, Prusner defined prions, infectious proteins. This goes back to the Kuru of Gaidusek. Doesn't have a nucleic acid, an infectious entity that does not have a nucleic acid.
Did he get a Nobel Prize?
He did.
Yes, he did.
He did get a Nobel Prize. Now, 1983, two big discoveries, which were just recognized by Nobel Prizes. The finding that HPV's human papillomaviruses cause cervical cancer by Harold Zurhausen, and the discovery of HIV by Montagnier and Barré-Sinoussi. We've talked about them.
Right, and others.
And others. 1989, Houghton identifies hepatitis C virus.
And that was, I think that was the first time that we had discovery from a commercial entity. That was Chiron.
It was a company, right?
Correctly.
And also it was cloning.
Yeah.
Probably using your technology.
Well, that's besides the point. But it's interesting how industry had absorbed all these very important molecular techniques and utilized them to solve this problem of what was the other agent that we clearly understood to be the predominant agent in the causation of hepatitis. Right. And it was missed for years, and nobody could find it, and nobody could isolate it. And this was a terrific discovery. And subsequently, it's been followed on in terms of trying to understand more about the virus. And we now have the first antivirals that are coming out. And so it's really been exciting. That and the development of the ability to propagate this in cell culture.
Right, which we have on our list, 2005. It took a while. The point here is that this was the first one discovered by cloning. Now, this is ushering in a whole new era of virus discovery, where we don't grow them, we clone them and sequence them. And
or we don't even clone them anymore.
We just sequence
now.
Just
sequence now.
All right. Because of
cheap sequencing.
Yeah, but there was an invention that allowed that to happen. Come on, guys.
What, sequencing?
Yeah, I mean, no, the polymerase chain reaction.
Actually, you don't even use that.
That's not used at all.
But that was a step along the way, though.
No, Sanger was a step along the way. Sanger and Wally Gilbert really laid open those fields, and modern technology is wonderful in terms of using molecular tools to end label bits of nucleic acid, and then using those end labels as primers for the synthesis and subsequent analysis.
I mean, we should mention that Sanger got a Nobel Prize. He got two, one for sequencing proteins and one for sequencing
I don't think
nucleic
he
acid.
published more than 30 papers, but I'd like to have been on any single one.
Aren't you glad you weren't on his faculty review committee that said, not enough publications, sorry, no promotion?
Well, he was at the MRC. Was he at the... Yeah.
Yeah,
I think he was at the MRC.
Yeah.
And it wasn't important. What was important was to do really, really incisive critical science, and that's certainly what he did.
All right, we're down the home stretch here. 1990, the first human gene therapy with a retrovirus vector, Anderson and Blaze. And here's one close to home, 1994, Kaposi's sarcoma virus identified by
That was
Chang
a lot
and
of
Moore.
fun.
They were here.
Yuan Chang was a fellow in pathology. Pat Moore was a research scientist at the New York City. He was head of epidemiology or something. Later came to public health, and they're sort of my scientific godchildren because I had the wonderful opportunity of helping them address many of the questions of what is the causative agents for this. And Yuan and Pat have taken that and run and identified this very interesting herpes virus that replicates in B cells, T cells. I can't even remember anymore, but it was also the causative agent of Kaposi's. And they've subsequently gone on to identify yet another virus using a novel technology that they developed, the Merkel cell carcinoma, which is a human papillomavirus. And we're seeing a lot of papillomaviruses in this discovery field that's coming out now.
1997, heart treatment for AIDS, highly active antiretroviral therapy using 3 drugs in combination to treat AIDS.
Beat them over the head because one isn't enough. The virus is just too good at getting out of the way.
And we have so many now antivirals that we can change the combination when resistance emerges. So you could live, if you can afford these drugs, you live a complete life. 2002 SARS outbreak and containment. It's a very interesting event. We haven't seen SARS since, but...
It was certainly exciting. The question is, was it much ado about nothing, or did we take it as seriously as it needed to be taken and do the right things? And I don't know the answer to that. I do know that killing all those chickens stopped the disease, the spread of disease, but...
don't forget the civets.
Well, finding out that the carrier was a small mammal.
Roof rat.
Well, now we believe
now
we believe that it originates in bats.
Bats.
And it infected civets and other animals, which were sold in the meat market.
So
and
it's
that's
the coronavirus paradigm.
But it was a dead end. I mean, several thousand people got ill.
Yeah.
It was very virulent. But
That's
then
the Chinese government. Well,
it
there was
died.
much ado about nothing, and they'll tell you about the $60 billion tourist industry loss that they suffered as a result of it. They don't think it was much ado about nothing.
Do you know the poster that Ian Lipkin shows in his seminar? It's a tourism poster for Hong Kong.
Yep.
Hong Kong will take your breath away.
Ian and I were both in Beijing talking about this as to how to control it as a public health outreach, which kind of stopped at the level of the Chinese middle management of the scientific community. But it was fascinating to see their take on it.
I think we've stopped it because we know the source, but you never know these days the virus.
I mean,
bat
it's a
viruses are
re-emerging
difficult.
infection, right?
You could find another way to get into people.
Exactly right, like Nipah virus.
So 2005, propagation of hepatitis C virus by Cesare, Rice, and Wakita. My understanding is the problem was they were all working on one serotype, and when they switched to a different serotype, which is what Wakita did,
Bingo.
it worked. And that was it. Sometimes that's what happens.
How many different kinds of hepatitis virus are there now?
Well,
A, B, C, D, E, maybe G.
What happened to F?
They didn't like the idea of an F virus.
Actually, there was an F, which was a DNA virus.
Oh, yeah?
If I remember correctly.
Okay.
I believe so.
2005, also the reconstruction of the 1918 influenza virus, Palazzi, Tumpe, and Taubenberger, using sequences from bodies exhumed out of the permafrost, pathology sections that have been kept since 1918. It's amazing.
Yep. Mind if I go back a little bit?
Yes.
In the year 1999, Ian Lipkin's group, then in California, sequenced the entire West Nile virus sequence out of 2 victims. That was the first time ever a viral complete genome had been sequenced out of a living tissue, or preserved tissue, I should say.
Yes, that's
So
also
that was
a
very
good one.
interesting.
Yep.
That should go in there someplace.
1999, that would be? Okay.
They were the ones that identified it as coming from Israel,
Right.
or at least the Middle East.
And finally, 2006, the HPV vaccine, human papillomavirus vaccine, which I think is an important vaccine.
So what's the controversy with that vaccine again, Vince? Just to recount our other, in the past conversations about this.
There are two vaccines. One is quadrivalent, and the other, I think, is trivalent, if I remember correctly. They are both based on making viral-like particles, I believe.
Right.
And they are specifically geared toward oncogenic papillomaviruses.
So you're going to prevent, what, cervical cancer?
You're going to prevent cervical cancer.
By giving this vaccine to who?
By giving this vaccine to young women.
Right. How young?
Well, that's a difficult question.
Prepubescent?
We want to do that then because we don't know when intercourse starts and how
does this
common.
give them permission to have intercourse if they're protected?
Not if they're my kid.
That's the controversy I was driving at.
But
you have a beautiful vaccine to prevent cancer in young women, and in giving it to very young women, it sort of says, you don't have to worry about that anymore. You can go ahead and have all the sex you want.
As you know, Dick, kids are going to do whatever they want.
They will.
So they might as well be protected. It's the same thing. The Catholic Church didn't want to use condoms to prevent AIDS because they said it implicitly says you can have sex anytime. I'm sorry, health comes first.
No, I totally agree with you.
Well, we all agree, but you're right. That's a good thing to point out.
Yeah, I mean, it's a social marketing dilemma.
Gentlemen, that stopped in 2006. I couldn't think of anything more recent. Anything come to mind?
Well, the new antivirals that are coming out against hepatitis C are very exciting.
They're not yet licensed, right?
They're not yet licensed, but they're real close, and the work has been very interesting, and they've targeted some of the same proteins, that is in terms of function as for HIV, and hopefully we're going to be able to use these specific virus targets for other virus diseases, although we know, for example, the incredible importance of proteases in morphogenesis and assembly of herpes viruses, and yet we don't have an efficacious agent that stops that and is useful, at least not yet.
Another issue that I'd like to bring up in this regard is that you guys have been focusing in on narrow zones of human virus health issues. And something else that's broken open in the last couple of years has been the discovery of a huge number of different kinds of viruses in the ocean, which gives lateral transmission of genes the option of going between two disparate species and therefore jump-starting evolution in different directions that you would hardly attribute to a Darwinian lineage type of thing, because the genes can now jump between an algae and a bacteria or a human from these viruses that are floating around in the ocean. And when you filter the ocean and then look for bacteria, you find very few. But they could still account for carbon, and they say, where the hell is this carbon? I can't see it. Well, a filterable agent, in quotes. You can't grow them either because their host species are no longer there.
Or we don't know what they are.
Right, so PCR would be one way.
You've just reminded me of something else.
I think that's a big discovery, by the way. I just wanted to add that.
Okay, that's a great point. We should actually talk about that in more detail sometime, because that's fascinating. That's fascinating.
The Czechs made a vaccine against pseudorabies virus, or herpes virus, and that was one of the first herpes virus vaccines. In fact, I guess it's the only one that's efficacious to any extent, and that was made with an attenuated pseudorabies virus to prevent infection of pigs.
Why is that called pseudorabies? Excuse me, I'm not well enough versed in this to know the answer to that.
And I'm not well enough versed in it to give you the answer.
But I guess we could ask them where your rabies virus is. Who, me? No, of course not.
Yeah, we'll have to have Lynn Enquist join us one day, because he works on pseudorabies.
And by the way, who discovered helper virus biology?
Oh, like viroids?
Yeah.
T.O. Diener. Yeah. He's on one of these lists. 1967, viroids. Oh, we can, we've mentioned it. It's important, yeah. The whole concept
they're interesting.
Yeah, they can't replicate on their own. They need a helper.
Right.
Yeah. There are lots of them in plants. Hepatitis delta virus is a human example.
Okay.
Adeno-associated viruses.
Exactly.
And what about all these jumping genes that we talk about? Were those formerly viruses, do you think? All these extra pieces of DNA that we've got in our genome that everybody keeps talking about as, I don't know what that is, Could those have been remnants of other viral infections that have accumulated in our genome and now find different purposes as the result of being there for so long? Or
I don't know.
what do you guys think about
Why
that?
not? Sure. I know we have lots of retrovirus remnants in
We
our
have
genome.
What if we
Huge
took out all of
numbers.
our introns,
Yeah.
right, and just put the genes together and said, "The hell with everything else. We don't need all that extra stuff." What do you think you'd end up with?
We wouldn't last very long, probably.
Because I mean, bacteria, they don't have that, right?
Yeah.
Viruses don't have that.
Well, that's not true. Viruses have introns.
Viruses
They
do.
have introns,
Sure.
Yes, they do.
but there are lots of organisms that don't have introns,
Mhm.
and they seem to get along pretty well.
It's a good question. It's
So when
a
you do a
very
frame
good
shift
question.
without splicing, you're dead, right? If you can't splice onto the other gene, you can't get rid of that big segment of DNA that doesn't have.
Well, one
Well,
idea is that they allow for modular evolution, right?
Yes. The other thing I
That's
wanted
the spliced pieces,
Sure.
not the introns, anyway.
are any viral genomes that you guys know about edited?
Sure.
Okay.
Absolutely. In fact, I believe editing was first discovered.
Not in
Yeah,
viruses.
measles virus,
it?
paramyxoviruses,
Oh,
yeah.
I thought it was.
Yeah.
in,
It's also done in a trypanosome. Now, gentlemen, put on your earphones and listen to a little clip from a podcast where they reviewed TWiV.
Uh-oh. I'm not sure I want to hear this.
This is my favorite link of the week because it's our podcast of the week, which I always love to do. And this podcast kind of made me laugh. I realized that sometimes I choose very strange podcasts, ones that are just a little more creative. This is from a scientist. He's actually a professor as well at Columbia University Medical Center. And it's a podcast all about viruses. All they talk about is viruses.
Wow. I
I
want to
know.
listen to that. I like viruses.
I know. Not computer viruses.
No. Virus.
Yeah, real virus.
Rhinoviruses.
Exactly.
Ebola virus. This week
Yes.
in virology.
I know. Isn't it great?
And it's on, by the way, it's on Ginger Campbell's sciencepodcasters.org site.
Cool, and I think they've done at least a dozen or so episodes so far. It's well produced, and it seems as though the person who sent us the email, Vincent, he loves doing the show, and it gives him a chance. He's been studying viruses for more than 30 years, so it gives him the chance to get together with other professionals and talk about viruses.
That's really neat. Well, we tried to get Ginger on the show, and she says, I was listening, but my Skype wasn't working, so we should
I
also
know.
get Ginger, because she has... With her, with this site, she has sciencepodcasters.org. She has aggregated some really good, including Mark Pelletier's Futures in Biotech, which is on this network, some really good podcasts on science. And I've been saying, again, another thing for my speech, it's good to go narrow. Much
Yeah.
better to do a virology podcast than to do a science podcast.
Oh, definitely. And this is narrow.
Narrow's great. I'm
I
much
know.
more interested in somebody who is passionate, crazy about one little slice of the world. That to me is more interesting than a generalist who just covers it all.
I totally agree.
Yeah.
So a fun little podcast of the week.
Oh, virology. Well, we won't play any of it. We don't want to make anybody sick, but you might want to listen. It's available through sciencepodcasters.org, along with a lot of other ones, including Dentalcast.
Yep. And it's also on iTunes as well.
Great. Oh, yeah. I mean, you
So you
got to
can
be
check
on
out
iTunes.
on iTunes.
If you're not on iTunes, you're missing out. And you can also
Missing
go
out.
to twiv.tv. He's got TWiV.
I know, that's why it made me laugh.
Vincent Racaniello and Dick Despommier. Very cool.
Very cool.
TWiV.
TWiV.
Yeah, they just do a dance.
Click and clap.
So that's amusing, right? So we're doing a good thing here, guys. Okay, we're going to wrap this up quickly. I have too many emails. I don't want to read all of them. Let me just read one, and then we'll do our picks and wrap it up. And we'll get to the other ones next week. What do you think, Dick?
Sounds good to me.
So Len wrote, you might be interested in knowing that Lily Pinio is alive and living with her sister in Florida. She's 91. He sent me a photograph. Lily was in the Lhasa incident.
She was the one that,
She was the nurse that was flown to the U.S.
raised the red flag, got medevaced out of northern
Right.
Nigeria by John Frame,
recovered
recovered and saved the life of
Jordi Casals. They used her serum.
Exactly.
So he said we should interview her. He'd like to know what she thinks of the book, what the author got right, what he didn't, and what did she do after it ended.
Right.
And this guy is apparently a history major, and is interested in the history of disease and medicine, and he thinks that the author of the book was too right there. He doesn't quite believe that. He says it makes me suspicious as a historian. So he'd like us to talk to her. So maybe we'll try to talk to her sometime.
Sounds like a road trip to me.
No. We're going to call her. We don't want to go to Florida. Okay.
Gotta go to Lhasa.
Let's wrap it up with our picks of the week. Science blog of the week, researchblogging.org. It's a very interesting site. It's basically a compendium of blogs which talk only about peer-reviewed research. Not chatting or rumor or anything, but you have to discuss a paper, and they all get posted there.
Club,
it's sort of like a journal club. It's a very broad range: biology, physics, chemistry, anthropology, neuroscience.
Great opportunity to learn.
So that's our blog of the week. Our science podcast of the week. Well, the Museum of Science in Boston does a podcast, and it's a very good museum. Any of you ever been?
Yes, I've been there.
And the podcast is nice. Really good science. It's weekly, a big range of topics, as you might expect. We'll put a link for that in the show notes. And our science book of the week, The Lives of a Cell.
Yes.
By Lewis Thomas.
Absolutely.
Great book.
You guys have probably both read it, right?
Yeah.
I've read it. It's fabulous. It's basically a collection of essays he wrote for the New England Journal of Medicine.
He was such a clever guy and so far-reaching and
Literate.
Not a great scientist in terms of his own science, but a tremendous ability to see what was important and how useful it can be, and a great leader, which is interesting. Science requires so much in so many different ways, and he contributed outstandingly in at least two, in terms of organization and in terms of patient care and public education.
Yep.
Yeah, these are really good. I mean, this guy was dean at Yale and NYU Medical, president of Sloan Kettering, a lot of high-powered positions, but these are very simple, very thoughtful essays about biology, The Lives of a Cell. Check it out. Okay, that should do it for another TWiV. Gentlemen, thank you. Saul, thanks
Always
for joining.
our
Dick,
Yep.
Alan couldn't make it today. He is writing articles on a deadline, but we'll see him next time. This is TWiV. You can send us your questions, comments. If we left someone out of this list, tell us what you'd like to hear. TWiV@twiv.tv. This is TWiV, This Week in Virology. Thanks for joining us. We'll be back next week, and you can find us on iTunes, by the way. Just search for TWiV. That should do it. Another TWiV is gone viral.